Thank you, IMP Vienna and the Max Birnstiel Foundation. It's a great honor to be here to receive that prize. It's also great to be back in Vienna because I did my master's here, so it's really fun to be back here in person and to give a proper presentation. During my PhD in Martin Pilhofer's lab, I was trying to image bacterial cell-cell interaction across several scales of resolution. And bacterial cell-cell interaction is actually quite important because a bacteria rarely occurs as an isolated cell in the environment, so as to steadily interact with other bacteria or other eukaryotic cells to ensure its survival. But bacterial cell-cell interaction can mean more. It could also mean, for example, how individual cells in a multicellular bacterium communicate with each other to, for example, split labor within this filament. But also eukaryotic cells develop strategies to actually block bacterial interaction and to hinder an infection. And during my PhD, I was actually able to work on these different aspects of bacterial cell-cell interaction. But today, I would like only to focus on one aspect, mainly how the most abundant protein in our urine protects us against a urinary tract infection. But what actually causes a urinary tract infection? More or less 90% of UTIs are caused by so-called uropathogenic E. coli, or short UPEC. UPEC are these hairy little bugs down here, so they are highly pileated bacterial cells. Pili are this hair-like extension on the bacterial surface, and in the case of uropathogenic coli, most of them are actually type 1 pili. And on top of the type 1 pili, so at the tip of the pillows, there sits FimH. And FimH can bind to high mannose type glycans. And this is really unfortunate for us because our complete urinary tract is covered with glycans because we have covered with uroplakins and they are highly glycosylated. So that means there are many handles for this bacteria to hold on to in our urinary tract. But our body developed an antagonist. And this is now the most abundant protein in our urine. This is called uromodulin. Uromodulin is produced in specific cells lining the kidney, and it's itself highly glycosylated. Once it's released to the urine, it forms homopolymeric filaments of roughly 2 micrometers in length. And as you can see here, in this previously taken negative stain electron micrographs, it has somehow a zigzag appearance. In the urine, uromodulin fulfills diverse physiological roles. For example, it protects us against kidney stone formation. It's involved in the activation of innate immunity. And what was quite interesting for us, it protects us as well against urinary tract infection. And the idea was, because it's a highly glycosylated protein, it competes with our uroepithelial cells for the binding of these pillows of this uropathogenic E. coli. However, the exact mechanism, how this works, and also the three-dimensional structure of uromodulin was so far unknown. And one reason was that actually uromodulin has quite a limited half-life in urine. So the whole project started when 2 of my colleagues at ETH, Max Sauer and Jessica Stanicic, in the lab of Rudi Klocksuber, were able to purify stable human uromodulin from urine. And the trick was just to add a bit of EDTA, because you always co-purify a metalloprotease, which naturally degrades uromodulin in urine. But this actually allowed us to now do a whole bunch of studies. And the first one actually was that together with Marcus Erbi's lab, we solved site-specific and comprehensive N-glycosylation map of uromodulin. So what you can see down here is the domain architecture of uromodulin. And on top, you can find these different types of glycans we found here, using like a glycomass spectrometry. We found several complex type glycans, but strikingly also 2 high mannose type glycans. And these are exactly the sites where FimH now, the tip protein of uropathogenic E. coli, can bind to. But we also wanted to incorporate these results on a glycosylation level onto a structural level. So that's why we took the purified uromodulin, plunge froze it on an electron microscopy grid, and used our core technique in the lab, which is cryo-electron tomography. For this, you transfer your sample into a transmission electron microscope and collect a so-called tilt series. This means 2D projection images of the identical feature, but later on you can take this projection image and calculate a 3-dimensional volume, which is our tomogram. And in this tomogram of the uromodulin fibers, we again saw the zigzag behavior, which was already reported previously, but we found a completely new orientation which looked more like a fishbone or, because it's almost Christmas, like a Christmas tree. When you have now several identical features in your tomogram, you can box out these subvolumes and average them together. This increases your signal-to-noise ratio. It's called subtone averaging, and this allowed us actually to resolve the first three-dimensional structure of human uromodulin. It has indeed a zigzag backbone, but it also has regularly spaced, laterally protruding arms. However, due to this intrinsic flexibility of the uromodulin filaments, our average remained at rather low resolution. So unfortunately, we couldn't map back any glycan set immediately. We had to use a trick. The trick was elastase. So elastase splits uromodulin in two parts. This part here in light blue actually gets degraded by elastase, and the part in green is elastase resistant. So when we now repeated our approach with only this elastase resistant part, we were only left with the zigzag backbone. And this allowed us now to come up with a model of the domain architecture of uromodulin, where the zigzag backbone is mainly composed of the so-called ZP domains, so they're called zona pellucida domains, and the arms were made of these domains in light blue. Actually, our domain architecture was then later the year confirmed by two studies, one from my colleagues at ETH, the other one from Karolinska Institute, where they solved the high-resolution structure of this filament backbone using single particle cryo-EM. But back for our study, this was enough to map back at least some of the glycans. We could say that one of the high mannose type glycans, so the binding partner of FIMH, was located on the uromodulin arm, whereas the other one was located on the uromodulin backbone. So when we now incubated uromodulin with FIMH, or only the lectin domain of FIMH, so the binding site, and again calculated a subtomal average, we found only an additional density on the uromodulin arm, where there was no additional density on the uromodulin backbone, indicating that only the high mannose site on the uromodulin arm is recognized. Further, biophysical analysis showed that actually 2 FIMH can bind to one uromodulin monomer, and this also fits to the volume of this additional density we found here in our subtomal average. When we then incubated uromodulin actually with pileated cells, we could indeed visualize how individual pili bind to this uromodulin, but one uromodulin, as is a filament, can bind to several pili. So that means these pileated cells got concretely encapsulated by uromodulin. And when you look at this at a bit larger scales, it also leads to bacterial clumping. And this was a complete new aspect of uromodulin biology, because it could mean that actually a bacterial clump is easier to remove with the urine flow compared to a single bacterium. Furthermore, if you add mannose to that sample, we couldn't observe any bacterial clumping anymore, which also indicates that this clumping is indeed mediated by FIMH binding to the glycans of uromodulin. Furthermore, it also means actually that we often treat UTI patients with mannose, and it could actually mean that we kind of hijack our own body's defense. When we now take these clumps, blanch freeze them again, thin them by a technique called cryo-focused ion beam milling, and again at cryotomograms, we found that these bacterial cells were completely embedded in a dense meshwork of uromodulin filaments. But the real power actually of cryo-electron tomography is to visualize macromolecular events in situ in an unperturbed sample. And this was more or less all in vitro. So that's why I set out and established a collaboration with the University Children's Hospital in Zurich. And our goal was to directly analyze patient samples with cryo-ET. I was working with doctors, and once they called me that I have a patient with a urinary tract infection, I took my bike, went there, took the sample, and directly went back and as fast as possible tried to process the sample. And with light microscopy again, found bacterial clumping. And strikingly, when we then took tomograms of the exact same unperturbed urine sample, we found pileated cells, and all were surrounded by numerous uromodulin filaments. In our tomograms, we could then reveal individual contact sites between pili and uromodulin directly in the patient sample. This was now from a patient suffering from a urinary tract infection caused by E. coli, but we also analyzed urine from patients with an infection caused by different pathogens, like Klebsiella, Pseudomonas, or Streptococcus. And to our surprise, actually we found a similar pattern. We again found bacterial clumping, and more or less 70% of these cells were also interacting with uromodulin. However, the exact mechanism is still not known. we have some hints that these other glycans we found in uromodulin can actually be recognized by other pili, so other pili type 1 pillars. Uromodulin competes with the binding of the type 1 pillars to the epithelial cells. As it's a filament, it's a multivalent binder which leads to encapsulation of bacteria and also aggregation. And these UG aggregates might be easier to remove from the urine with micturition. And with this, I'm at the end. I have to thank my supervisor, Martin Pilhofer, who gave me lots of freedom in exploring really different kinds of bacterial cell-cell interactions. Also would like to thank the Pilhofer lab, who created an awesome working environment. This was where I presented really a collaborative study with Rudi Glocksuber's lab, especially Max Sauer and Jessica Stanisic, and also Markus Erby, who did the glycomass spectrometry. And the study was actually a bit the start of my own little subgroup, where I also have now established in Martin's lab, and we are focusing how we actually can further harness clinical samples for cryotomography studies. And with this, I'm at the end, and I want to thank you again for your attention, and happy to take questions.